Dear students, uh, from genetics we are into one more topic, that is mutations. So this is a topic related to mutation. We all know mutation is a change. So if you see here, a heritable alteration or change in the genetic material is called as mutation. And if you see, there are different types of mutations. If you see here, this is the original chromosome, this slide which includes the image. And if you see here, the alterations, how it is being done here. If you see here, A, B, C, and these are two pairs, that is D, E, F. Here if you see the deletion, you can see here from this it is formed as AC and DEF. Here you can see the deleted one, that is the B. And if you see the duplication here, you can see here the B has been in a doubled, it's a duplication. And the other type, if you see, is translocation. What does translocation means? So if you see here, A, B, C, J, K, L. And if you can see here, you have the other one, that is the other one added, that is G, H, I, D, E, F. You can see it's a multiple types. And the next one is inversion. Inversion in the sense is that if you can see here, it has been reversed or inversion. If you see, this is the one original one, and here if you see AEDCBF. So these are the different types of mutations. So this includes the different types of mutations and its effect, how it is. Just if you see, these are the types. Next, if you see here the substitution mutations, if you see here the DNA, mRNA, amino acids, and if you see when it undergoes the substitution, the target, if you see here DNA, how it has been, if you see the starting ones, all the elements here, how it is been substituted by this T. Next, mRNA also, if you see here, how it has been substituted here, okay, with adenine. So amino acids also, if you see here, the changes. So substitution, so what is substitution? A substitution is the replacement of a single nucleotide by another. So most common type of mutations, if substitution involves replacement by the same type of nucleotide. If you see here, there are two types: transition, transversion. Translation is more frequent. That means a pyrimidine P for a pyrimidine, C for T or vice versa, and a purine for a purine, A for G, adenine or guanine, or etc. Transversion is a pyrimidine by a purine. That means it has been transferred. It is not same. A pyrimidine by a purine and a purine for a pyrimidine. So this is about substitution. Substitution also includes the mutations, which includes the point mutations. Here, this is a diagram which showing the point mutations. Here, if you see the genetic material, the DNA, how the DNA and the mRNA, which is into amino acid. So deletion, if you see, loss of one or more nucleotides is called as deletion. Small deletions may lead to frame shift. Larger deletions may result in partial or complete gene deletions, or sometimes unequal crossovers. So this diagram shows the insertion, what I already told you all in the previous one. So, what is insertion? An insertion involves the addition of one or more nucleotides into a gene. Small insertions may lead to frame shift. So, this is a frame shift where you can see how it has been inserted here. If you see, the first one and the second one is not as similar. If you see how here it has been inserted here, okay, how the guanines have been added here. So chromosomal, this picture illustrates the chromosomal rearrangements. That means translocations, how the translocations, deletions, inversions, all that what I have explained, how it has been changed, how it has been divided into fragments, everything, how it is being taking place has been done here. So this one represents the chart of trinucleotide, how it has been repeated. You can later take the screenshot and find out what it is. Next, if you see here, this also includes the trinucleotide repeats, which includes this is extra information, students. Just I have given you all this image so that you will be knowing more for a future studies. You can afterwards ask me regarding this whenever you require, how this trinucleotide had been repeated, as I told you all, how it been takes. So polymorphism. So what is polymorphism? Sequence variants with no obvious effect upon phenotype may be termed as polymorphism. That means sequence variants are there with no effect upon the phenotype may be termed as polymorphisms. So structural effects of mutations. So there are different structural effects of mutations. There are non-synonymous mutations, synonymous mutations. Non-synonymous mutations include missense, nonsense, frame shift. So, if what is non-synonymous mutation? If a mutation leads to an alteration in the encoded polypeptide, it is known as non-synonymous mutation. So, what is this missense mutation? Here, in this case, a single base pair substitution can result in coding for a different amino acid. And the synthesis of an altered protein is called as missense mutation. And what is this nonsense? A substitution that leads to the generation of one of the stop codons will result in premature termination of translation peptide chain is termed as this nonsense mutation. That means premature termination will take place here. Frame shifts. Frame shift mutations is if a mutation involves the insertion or deletion of nucleotides that are not a multiple of 3. It will disrupt the reading frame and constitute a frame shift. While the frame is being reading, it will be disrupted by constitution of frame shift mutation. That means if a mutation involves the insertion or deletion of nucleotides that are not of a multiple of 3 is called as frame shift mutations. So mutations in non-coding DNA. In general, mutations in non-coding DNA are less likely to have a phenotypic effect. Exceptions include mutations in promoter sequence or other regulatory regions or the affect the level of a gene expression. So mutations in mRNA and siRNA are binding sites likely to result in the disease. So, what are the different causes for mutations? You see spontaneous mutations, mistakes in replication, induced mutations caused by a mutagen. So, if you see induced mutations in this mutagens, chemical and physical. So, radiations also can cause mutations. If you see this diagram represents the radiation which cause the mutations, electromagnetic, ionizing X-rays, microwave radar, television, radio, cosmic gamma rays, this wavelength which will include the radiation which can cause the mutations, effects of UV radiation. So if you see the UV radiations, how will affect, if you see basing pairing on opposite strand does not occur here. If you see the diagram here, polymerase skips during replication. Unfortunately, these are repaired actually. So effect of how the UV ultraviolet radiations on DNA structure. If you see the before one, when if you see in this diagram how the UV photon affecting, after the affecting, how it has been this disruption here after in this diagram. If you see here. Now, ionizing radiation and any radiation capable of displacing electrons from atoms or molecules, thereby producing ions, are called as ionizing radiation. Can cause breaks in the phosphodiester backbone. DNA cannot be replicated unless repaired. Some examples are alpha, beta, gamma, and X-rays. Causes formation of free radicals and toxic oxygen. Next, if you see here, so these are the radiations What I have explained, different radiations and different effects which can be caused in the mutations, and I also explained about different types of mutations, how it has been affecting. Okay, students, this is all about mutations. Thank you.